Chapter 9 of your molecular textbook covers gene mutations. These include deletions, insertions, inversions, translocations, and other changes that can affect one base pair to hundreds or thousands of base pairs. These gene mutations are a change in the nucleotide sequence in a particular gene. They're slight structural alterations. In this chapter, we'll be discussing a comparison of phenotypic consequences of point mutations, distinguishing detection of known mutations from scanning for unknown mutations, discussing methods used to detect point mutations, and describing mutation nomenclature for expressing sequence changes at the DNA, RNA, and protein levels. There are various types of gene mutations that can occur, but it's important to remember because there is more than one codon for most of the amino acids, DNA sequence changes do not necessarily change amino acid sequence, and this is an important concept for interpreting results of mutation analysis. Silent mutations involve substitution of one nucleotide with a different nucleotide that does not change the amino acid sequence. Conservative substitutions may change the amino acid sequence, but the replacement and the original amino acid have similar biochemical properties, and the change will not drastically affect protein function. In contrast, non-conservative substitution results in the replacement of an amino acid with biochemically different amino acids. A nonsense substitution mutation terminates proteins prematurely when a nucleotide substitution produces a stop codon instead of an amino acid codon. Insertion or deletion of other than a multiple of 3 nucleotides results in a frameshift mutation, throwing off the triplet code out of frame. The amino acids in the chain after the frameshift mutation are affected, as the triplet code will include new combinations of 3 nucleotides. Point mutations are alterations of a single or a few base pairs that have a range of effects on proteins. Non-conservative, nonsense, and frameshift mutations will generate different phenotypes depending on where they occur along the protein sequence. Point mutations in the end of a coding region may have minimal consequences, whereas mutations at the beginning of a coding sequence are more likely to result in drastic alterations or even effective deletion of the protein coding region. Shows examples of DNA sequences exhibiting the different mutation types and the effects, if any, on the amino acid. In the first row is a normal DNA sequence with the respective adenine, thymine, guanine, and cytosine bases. On the second line, you have an example of a silent mutation. So you see here the guanine is substituted for a thymine, but it has no effect on the amino acid sequence. In the third row is a conservative mutation in which the change in bases has changed the amino acid sequence, but even so, the amino acid that has been replaced is one that has similar biochemical properties. So in this example, leucine has been substituted for valine, which have similar properties. The next example is a non-conservative mutation in which the base change has resulted in a alternate amino acid, proline, which does not have similar biochemical properties. The second to last line shows a nonsense mutation in which the protein is terminated prematurely, and the last shows a frameshift mutation in which the change in base has thrown off the triplet code out of frame, resulting in a different amino acid sequence. Guidelines for human gene nomenclature were first published in 1979 when the Human Gene Nomenclature Committee was first given the authority to approve and implement human gene names and symbols. Standardization includes gene names needing to be capitalized and set in italics with no hyphens. Protein names are not italicized or completely capitalized. So on this slide, there's a couple examples of gene nomenclature. So the top example shows a nucleotide mutation sequence 5162, which is the base location, with the original base of G being replaced by A. And this is expressed as shown with the base position with the original base immediately after it, then a greater than symbol, and the base that has replaced it. The bottom example shows an amino acid with Q being the one-letter abbreviation for the original amino acid. The abbreviation Q is actually that for the amino acid glutamine, in which the possible codon could be CAA or CAG. The amino acid position is 197, and it's being replaced with G, which is glycine, as a result of the mutation. Insertions and deletions are respectively named with the position of the mutation with the words INS or DEL, with INS indicating insertion and DEL indicating deletion. They're immediately followed by the replacement base or protein, depending on the mutation. So although standardization has been set forward, it's still very confusing, and misnaming can be found in various studies. Various detection methods exist for mutations that include biochemical methods such as immunoassays, high-performance liquid chromatography, or mass spectrometry, along with hybridization-based, sequencing-based, and cleavage-based testing. For our purposes, we will not be able to go through every single method in detail, but we'll focus on a few in each category. Biochemical methods are used to directly analyze the change in protein structure or function instead of identify the point mutation itself. Biochemical methods include those such as immunoassays that use specific antibodies or ligands to detect presence of target molecules, HPLC, or high-performance liquid chromatography, that can be used for separation and analysis of amino acids, and gas chromatography used to detect drugs, poisons, and their metabolites in clinical samples. HPLC differs from gas chromatography. As the name implies, it consists of two phases, a column with a mobile phase or a solvent that flows through a stationary phase or solid support. Gas chromatography involves a mobile phase of inert gas and a stationary phase of a high boiling point liquid absorbed to an inert solid support in a column. In HPLC, the migration rate of molecules is measured, whereas in gas chromatography, the strength of the interaction or dissolution of the sample components into the liquid phase results in varying retention times in the column, and these are monitored by a detector to be measured. Mass spectrometry is another biochemical method of analysis. In mass spec, an ion source sends high-energy electrons that hit the target sample molecules, separating into ions. Usually these are cations, which hold a positive charge, with loss of 1 or 2 electrons. The ions themselves may further fragment into smaller ions and neutral particles. This collection of particles is accelerated and focused into a beam through a magnetic field. The ions are deflected by the magnetic field according to their mass and charge, and by varying the magnetic field strength, different ions are aimed at a detector. The readout of the instrument is a spectrum with the mass charge value on the x-axis and the abundance of ions on the y-axis. Various ionization methods can be employed, including electrospray ionization, which involves a test sample converted to a fine spray of charged droplets that are measured. MALDI, in which ions are produced by firing laser pulse into surface coated with material that generates a positive ion from a peptide. A high positive charge on the plate surface repulses the ion, which flies to the detector. And MALDI-TOF, matrix-assisted laser deabsorption ionization using time of flight for measurement, in which high molecular weight molecules are identified using this method. And this is a method that I've also mentioned that is currently being used for microbiology identification as they identify organisms based on their protein structure. One other ionization method is CELDI-TOF, which is surface-enhanced laser deabsorption ionization. CELDI-TOF is just an extension of MALDI used to measure peptides. CELDI normally uses a chip with a chromatographic surface, making the purification of the sample implicit. For MALDI, the purification needs to be done before application to the plate. Hybridization-based methods are the next group to discuss that are used for detection of gene mutations. We've already discussed or touched on some of these methods, but we'll go into a little more detail of some of the methods, such as single-strand conformation polymorphism, allele-specific oligomer hybridization, melt curves, and array technology and their applications on the following slides. For single-strand conformation polymorphism, dilute concentrations of short double-stranded PCR products are denatured, followed by rapid cooling. Because the diluted single strands cannot easily find their homologous partners, they fold by intrastrand hybridization, forming three-dimensional conformers. The shape of the conformer depends on the complementary nucleotide available. A single base difference can cause it to fold differently. The conformers are then subsequently resolved in polyacrylamide gel or by capillary electrophoresis with strict temperature control. The speed of migration depends on the shape and the size of the conformers. SSCP can be sensitive enough to detect mutations in samples containing as low as 5% potentially mutant cells. This diagram shows an example of a normal control and test sample with a mutation in which single-strand conformation polymorphism is performed. Firstly, the region of interest needs to be amplified using PCR. The PCR products are then denatured and diluted. And lastly, the conformers, or the single-stranded folded DNA, is separated using either page or capillary electrophoresis. After the conformers have been resolved using electrophoresis, as seen on the previous slide, the results can be analyzed against the control to determine if mutations exist. On the polyacrylamide gel sample on the left, you can see the normal control in the first lane. The dotted line mutations appear in the middle lane, and the last lane in which both the normal control DNA is present along with the mutation, indicating the patient is positive for the gene mutation. The capillary gel depicted in this right figure shows the same results just in peak forms versus the bands seen on the gel electrophoresis. Patterns are detected using silver stain, radioactivity, or fluorescence. So here's just another example of a figure using PAGE, in which T1 is the test sample without the mutation, T2 is the test sample with the mutation, and NC is the normal control. Of note, ethidium bromide stain is not used because it's less sensitive than the other stains or methods. Allele-specific oligomer hybridization is a dot plot method of hybridization similar to the southern blot method that uses an immobilized target and a labeled probe in solution. The ASO procedure begins with amplification of the gene region of interest by PCR, similar to single-strand conformation polymorphism. After the PCR product is spotted onto a nitrocellulose or nylon membrane that are soaked in a high salt denaturation solution, the DNA on the membranes are neutralized with dilute acid and fixed to the membrane. Labeled probes matching the normal and mutated sequences are then hybridized to the membrane in separate reactions. Allele-specific oligomer hybridization has been used in clinical testing for detection of specific mutations and polymorphisms and typing of organisms and tissue typing as well. It's also been carried out as a reverse dot plot in a 96-well plate format for infectious disease testing for organisms such as chlamydia, trachomatis, and mycobacterium tuberculosis. Here show an example of allele-specific oligomer hybridization. So 3 samples are spotted on 2 membranes. One membrane is probed with a labeled oligomer of the normal sequence, indicated on the left as this positive probe, and the other with a labeled oligomer containing the mutation, the M probe, indicated on the right. A normal sample, indicated as positive, hybridizes with the normal oligomer only. A homozygous mutant sample, indicated by MM, hybridizes with the mutant oligomer only, and a heterozygous mutant sample, indicated by M/positive, hybridizes with both oligomers. So this figure on the left shows a probe complementary to the normal sequence, whereas the right shows a probe complementary to the mutant sequence. This example shows autoradiograph results of allele-specific oligomer hybridization using chemiluminescent detection. One normal sample and one heterozygous sample are shown with a heterozygous mutant control, a normal control, and a negative control. So here you can see in lanes 1 and 2 are patients in which 1 is a normal patient, 2 is a heterozygous with a mutant and normal probe detected. M is the heterozygous mutant control, and plus is the normal control, and negative has no probe detection since it's the negative control. Melt curve analysis is a post-amplification step of real-time PCR. PCR amplicons generated in the presence of DNA-specific fluorescent dye are heated at a specific rate. As can be seen from the name of this method, it's based on melting temperature and can be performed with or without probes, but it does require dyes. Non-specific dyes can also be used to screen for sequence differences, but since not sequence-specific, they do not distinguish between the target amplicon and extraneous products in the PCR reaction. Of the DNA-specific dyes that can be used, ethidium bromide, Cyber green, or LC green can be used and are heated at a rate of about 0.3 degrees Celsius per second. Specificity of melt curve analysis can be increased by using high-resolution melt curve analysis, which uses fluorescent resonance energy transfer, or FRET probes, that hybridize next to one another across the sequence position being analyzed. The probes fluoresce only when bound to the target sequence because FRET fluorescence relies on the transfer of energy from a donor fluorescent molecule on one probe to an acceptor fluor on the other probe. With melt curve analysis, the dyes specific for double-stranded DNA initially yield a high signal because DNA is most double-stranded at low temperatures, but as the temperature increases, the double-strand DNA separate into single strands, losing the dye. The fluorescent signal gives a pattern, as seen in this graph, where the fluorescence decreases as the temperature rises. As can be seen in the graph, and from what has been discussed previously regarding stringency, every different target sequence has a characteristic melting temperature, so analyzing the melt curve indicates which sequence is present. In this example, homozygous normal, heterozygous, and homozygous PCR products are analyzed and display the varying curves due to the differences in the target sequences. Instrument software subsequently plots the derivative of the fluorescence data versus temperature to show the melt curve as peaks at the melting temperature of the test sequence. A normal homozygous sample should have a TM that can be distinguished from that of a mutant sequence. In this example, you can see that the mutant is shown in red compared to the normal sequence showing one larger peak in black. The next method we'll discuss is array technology. So this includes methods such as reverse dot plots, a method we've discussed previously that involves multiple probes spotted on a membrane that are all exposed to the same target. Array technology is used to investigate multiple genomic sites at the same time. Array technologies include macroarray, microarray, high-density oligonucleotide arrays, and microelectronic array methods. Microarray technologies include comparative genomic hybridization, which can either be micro or macro arrays, expression array, and SNP detection, mutation analysis, or sequencing, which utilize high-density oligonucleotide array technologies. So we'll discuss high-density oligonucleotide arrays in a little more detail. Mutation analysis, such as that for the p53 tumor suppressor gene, can be performed using array technologies such as high-density oligonucleotide arrays. This method can interrogate thousands of genes simultaneously. It does require a new array for each sample, and it involves unlabeled probes that are synthesized on the substrate. Arrays such as high-density oligonucleotide arrays are designed to test multiple genes for sequence mutations. So to do this type of analysis, the test DNA is fragmented by treatment with DNase before binding to the complementary probes on the array. If sample fragments are too large, such as in samples not treated with DNase, a single base pair mismatch has minimal effect on hybridization, so the fragment binds to multiple probes, and the specificity of detection is lost. An example of one type of hybridization format is known as tiling, or standard tiling. In this format, the base substitution in the immobilized probe is always in a certain location from the 3' end. Tiling uses overlapping probe sequences to blanket detect nucleotide changes in the sample. After hybridization of the fluorescently labeled sample DNA, The fluorescent signal is read on a scanner with appropriate software, and the mutations are identified as indicated by which probes are bound. This method is not routinely performed in clinical laboratories, but a number of applied methods have been developed using the high-density oligonucleotide and microelectronic arrays. Shows an example of mutation analysis by high-density oligonucleotide arrays, in which a heterozygous mutation has occurred. The normal nucleotide sequence, as displayed in this top figure, is TCG, whereas the bottom sequence shows that cytosine is replaced with adenine. Each sequence position is represented by 10 spots on the array, 5 sense and 5 antisense probes. The sequence binds only to its exactly complementary probe. Binding of the sample fragment in high-density oligonucleotide array is detected by increased fluorescence. The next group of methods we'll discuss are sequencing or polymerization-based methods that include sequence-specific PCR, also known as SSP-PCR, allelic discrimination, and direct sequencing. Sequence-specific primer PCR is commonly used to detect point mutations and other single nucleotide polymorphisms. There are a number of modifications to the method that involve design of primer so that the primer 3' end falls on the nucleotide to be analyzed. Successful amplification will occur only if the 3' end of the primer matches the template. By designing primers to end on a mutation, the presence or absence of product is interpreted as the presence or absence of the mutation. Sequence-specific primer PCR is routinely used for high-resolution HLA typing and for detection of commonly occurring mutations. This figure provides an example of a multiplexed SSP PCR in which the mutation in the BRCA1 gene is detected by an allele-specific primer that ends at the mutation. Of note, the BRCA1 gene provides instructions for making a protein that acts as a tumor suppressor. Tumor suppressor proteins help prevent cells from growing and dividing too rapidly or in an uncontrolled way. So mutations in this gene are associated with various cancers, such as breast or pancreatic cancer. So in this example of SSP-PCR, mutation lanes are shown in the two middle lanes designated by M, in which you can see primer products specific for the mutation. So these are the extra bands present that are not seen in the normal lanes. Allelic discrimination is another format that uses fluorescently labeled probes. This method is similar to the TACMAN technology that has been discussed previously. In allelic discrimination, color is generated for mutant or normal sequences. Allelic discrimination can be performed on real-time PCR instruments, and an advantage of this assay is that multiple samples can be analyzed simultaneously, and it has been proposed as a practical high-throughput laboratory method. In allelic discrimination, probes complementary to either the normal sequence, shown in green in this figure, or the mutant sequence, shown in red, are labeled with different fluors, such as the FAM or VIC fluorescent dyes. The signals are detected and analyzed by instrument software, And as mentioned previously, multiple samples can be analyzed simultaneously. The Taq exonuclease functions only if the probe is matched to the sequence being tested. High FAM fluorescence indicates a normal sequence, whereas high VIC fluorescence indicates a mutant sequence, and as if both fluors are detected, such as here in the figure, the red and the green fluorescence, the test is considered heterozygous. The next group of methods we'll discuss are cleavage-based methods, and these include restriction fragment length polymorphism, also known as RFLP, nuclease cleavage, and invader technology. If a mutation changes the structure of a restriction enzyme target site or changes the size of a fragment generated by a restriction enzyme, RFLP analyzed can be used to detect the sequence alteration. In the figure, the normal sequence seen in the top line above the electrophoretic pattern is converted to an ECO-R1 restriction site by a G to A mutation. The presence of the mutation is detected by testing the PCR product with the respective enzyme. The agarose gel panel shows the predicted gel patterns for the homozygous normal, seen here on the left of the agarose gel, the homozygous mutant, seen in the middle, and the heterozygous sample. Each is also shown as an uncut fragment and a cut fragment. Several PCR RFLP methods are widely used for detection of commonly occurring mutations, such as factor V lead-in, and it's also been used for HLA typing. The detection sensitivity of heteroduplex analysis can be increased by using single-strand specific nucleases that cleave heteroduplexes at the mispaired bases. The region of interest is then amplified by PCR, and the PCR product is denatured and renatured with or without added normal PCR product. Renatured duplexes are then digested with nuclease, and the products are observed by gel electrophoresis. In this figure, heteroduplex analysis begins with PCR amplification, in which heteroduplex formation occurs with the mutant sequence. The mutation on the target DNA can be seen as the pink dot in this diagram. As mentioned on the previous slide, once the PCR products are obtained, they are then denatured and renatured to move forward with the single-strand specific nuclease format. After PCR, the single-strand endonucleases cleave mispaired regions of heteroduplexes, then the cleaved fragments can then be resolved by agarose gel electrophoresis. The normal can be seen in the first lane after the molecular weight marker designated by M, and all subsequent lanes show mutants in which various cleaved fragments can be seen, indicating the presence of mutation. The last method we'll discuss is invader technology. This assay is a method developed by third wave technologies, or Hologic. It's based on characteristic enzymatic activity of their proprietary enzyme named cleavase. So this technology involves mutation detection in which the sample is mixed with probes and enzyme. The enzyme cleavage of the probe test sample hybrid will yield a fluorescent signal, and the signal will only occur if the probe and test sample sequence are complementary. So with Invader technology, the pre-mixed reagents include both probes and enzyme. The reagents, including cleavase, are added to a 96-well plate along with the test samples. The samples can either be DNA or PCR products. Hybridization of supplied probe and anchor sequences to the input 10 plate, as seen in the upper left image of this figure, forms a structure that is the substrate for the cleavage enzyme. The enzyme removes the flap sequence, which form another hybridization structure with labeled probe. The second cleavage releases the fluorescent dye from the vicinity of the quencher on the probe, and the fluorescent signal is then detected. If the template does not match the probe in the first hybridization, no cleavage occurs, and then no signal is subsequently detected. A couple important points to remember that were covered in this chapter include that mutations and polymorphisms are changes in the DNA sequence. DNA sequence changes have varying effects on the phenotype, as we saw when we discussed the terms such as silent mutations, substitutions, frameshift mutations, and point mutations. The molecular detection of mutations include various techniques and methodologies, including hybridization, sequence, or cleavage-based methods. So some of the important ones we discussed in this chapter include single-strand conformation polymorphism, or SSCP, allele-specific oligomer hybridization, melt curves, and array technologies, which all fall under hybridization methods. Sequence methods include sequence-specific PCR, such as SSP-PCR, or allelic discrimination. Cleavage-based methods include RFLP analysis, nuclease cleavage, and invader technology. And finally, another important method of molecular detection includes biochemical methods that directly analyze the change in protein structure and function rather than identifying the point mutations themselves. So an important method that you should be familiar with is MALDI or MALDI-TOF.